This video describes the usage of the GUI or Graphical User Interface version of the GlycoPAT software run at the MATLAB interface. The operation of the standalone version of GlycoPAT is very similar, and this is also briefly described in this video. Here we will show how to 1. Install the software in MATLAB, and 2. Run basic GUI functions. More detailed user instructions and examples are provided in the full GlycoPAT user manual that is available with the software. To install the full GlycoPAT software, simply copy the GlycoPAT software folder to a desired location like C/GlycoPAT. Open the MATLAB program and browse to this GlycoPAT folder. Then type install GlycoPAT at the command prompt and press enter. This initiates software installation and compatibility verification. Next, change the user directory to the folder containing the program input. In this case, it is the demo subfolder. To initiate the GUI, type GlycoPAT GUI at the command prompt. This opens a window with 5 different options or functions. Glycopeptide digestion. This enables the creation of a search library or database with a series of glycopeptides that we are interested in looking for. Tandem MS analysis. This enables the matching of specific experimental spectra to members of the glycopeptide search library generated in the previous step. Browse results. This is a viewer to analyze the results from the previous step. Glycopeptide fragmentation. This enables fragmentation of glycopeptides and generation of a list of possible fragments. Single spectrum annotation. This provides an interactive interface for the detailed analysis of a single spectrum, rather than the tandem MS analysis step described above, which performs such analysis for an entire MZXML file under a constant condition. Here we demonstrate the first 3 functions. Usage of other functions follows, and details can be found in the full program manual. First, click the Digest button. This opens the Digest GUI that is used to create the glycopeptide database. This window requires several inputs, including a protein sequence in FASTA format. This is just a regular FASTA file, and in this case, a single protein containing human fibronectin. Specification of an enzyme among a choice of varieties, trypsin in this particular case. Specification of fixed modification file, which includes the modification of cysteine by iodoacetamide indicated by the transformation of C to Ci. Specification of variable modifications. In the current example, this file includes 9 different potential ending glycans depicted in the SGP 1.0 format. This can be expanded readily by including additional members also in the SGP 1.0 format. Specification of miscleavage, maximum minimum peptide size, maximum number of variable modifications, etc. Once the inputs are provided, press Digest. This generates a list of candidate glycopeptides. The top section has the input parameters used to generate the database, and the bottom section provides the members of the glycopeptide database. These data can be saved onto the hard drive for use in the next step. In the next step, open the window for analysis of MS data. Here, the glycopeptide database is first loaded. The output location and file name is specified, and additional parameters are changed as necessary. The input experimental MZXML file is also selected. The nature of fragmentation is set to auto since the MZXML file already contains the fragmentation mode data. More details on other parameters and analysis options are provided in the full program manual. Pressing the Score All file results in the search of the experimental data to find the members of the glycopeptide database. Information on the scan number being analyzed and potential glycopeptides identified appear in the command line of the analysis program and also in the small window. Similar to the current GUI version that is run in the MATLAB environment, a standalone GUI version of the program is also available that can be run in an identical manner, yielding exactly the same results. While this standalone version is not shown here, its installation is straightforward. Simply download the program from www.virtualglycome.org and double-click the program to start it. This standalone version does not require the MATLAB software program, and it is platform independent. The MATLAB version is shown here since it allows a more complete explanation of the workings of GlycoPAT. The analysis takes a few minutes. Once the analysis is completed as indicated, we are ready to browse the results. Loading the output file from the previous step brings up two tables containing the header information and the results. In the results, the final findings are arranged in order of descending ensemble score values, and there are also additional parameters provided. Next to the column of ensemble scores is a column that provides the decoy ensemble scores for scrambled glycopeptides. If you're only interested in spectrum below a particular false discovery rate, this can be set at the bottom of this GUI, either for peptides, glycopeptides, or a combination of these. Applying this SDR filter results in an ensemble cutoff score which describes the minimum score required for a good hit. Additional options are also available to trim the results based on the user needs. To view the resulting spectrum, click on the corresponding row. This results in the program reading the input MZXML file, reanalyzing the score files again, scoring the file again, and finally displaying the experimental data with matched peaks shown in green and unmatched peaks shown in red. Zoom in to a particular portion to view these peaks more closely. You can see the exact masses of the peaks and also the nature of the fragmentation based on the conventional BY nomenclature. Individual files can be saved in a variety of formats in this particular window. All files that are analyzed can also be automatically annotated and stored in a particular folder using options provided in the Browse Results window. This requires additional installation of a freeware called GoScript. We'd like to thank you for your interest in our project.